Thank you so much. I think it's been a great 1.5 days talking to you all about various different aspects of unmet drug development needs of neonates, patients with alpha-1 antitrypsin deficiency, type 1 diabetes, lysosomal diseases, really intellectually stimulating discussions, questions from the audience, comments from the audience. It's been great. This is the last session of the day, and I'm super excited to get started with this. Even though the topic of this last session is considerations for clinical trial designs in neonates and type 1 diabetes, but the specific issues that we are going to discuss here, I'm very sure are going to be equally applicable to our stakeholders in lysosomal disease community and alpha-1 antitrypsin deficiency community, because for the past couple of days after speaking with our stakeholders, we've heard that some of the things that we want to discuss, they also want to learn from those areas, because we're going to be talking about, things like decentralized clinical trials, external controls, and how to make conduct of clinical trials more efficient. This is an issue and challenge that everybody in the drug development community faces. So yes, it is officially the title is neonates type 1 diabetes, but broadly applicable to pretty much any other drug development area that we might want to be talking about here. So with that, I'm going to invite our panelists to the stage. My co-moderator, Joe Hedrick, who's the Executive Director of Type 1 Diabetes Consortium. As the panelists, I'm really honored to have AJ Allen, who's the Chief Medical Officer of IAC forChildren, Ralph Bax, who's the Head of Pediatric Medicines at the European Medicines Agency, Dr. John Concato, who is the Associate Director of Office of Medical Policy at CEDAR, and he's joining us from online Zoom. Dr. Anne Massaro, who's the Supervisory Medical Officer at Office of Pediatric Therapeutics. Tom Miller, who is the Vice President of Acute Chronic and Pediatric Disease Nucleus at Bayer Pharmaceuticals. Victoria Niklas, who's the Chief Medical Officer at Oak Hill Bio. Betsy Pilon, who is the Executive Director of Hope for Hypoxic Ischemic Encephalopathy Organization. She's a long-standing INC parent stakeholder, and she's also joining us on Zoom. Then we also have Dr. John Sharretts, who is the Director of Division of Diabetes and Obesity. I'm sorry, I'm not going to misspell it, but it is Director of DDLO at FDA CEDAR. And similarly, we also have Dr. Justin Penson-Statler, who is the Lead Clinical Analyst at CEDAR. Honored to have you. Yes, sorry. Honored to have you. And thank you so much. So we can just get started. Before I started, I just wanted to make sure that Dr. Concato is online. He's able to hear us. Are you getting us, Dr. Concato?
I can hear you well. I hope you can hear me. Regrets for not being able to be there in person. I'm overseas for a different meeting, but a privilege and a pleasure to be here.
Thank you.
Zoom.
Thank you so much. Really appreciative that you were able to find some time to talk to us about issues related to clinical trial design. And similarly, just checking with Betsy. Betsy, are you hearing us? And if you are, can you please speak so we know that you're here?
Yeah, thank you so much for welcoming this group to the conference. I'm excited to be here and share the neonatal patient family perspective.
Awesome, great. And as you can see from the number of panelists that we have on this session, how important this topic is to a wide variety of and diversity of the stakeholders that we work with on a basically pretty much daily basis. So let's just get started with this session. And I'm actually going to pause the very first, just going to get started right onto the business and talk about one of the most important aspects that we wanted to have a discussion about. That is regulatory aspects about usage of external historic controls, evaluating the critical regulatory considerations for designing clinical trials that may want to use external controls, talking about the role and reliability of those controls in enhancing the validity of the trials. I mean, officially neonates and type 1 diabetes, but definitely, as I said before, heard from our alpha-1 and lysosomal disease community. They also wanted to have a similar sort of a discussion there. So enhance kind of a discussion about it. So I will pose this question to Dr. John Concato, who we all understand that he's the, one of the experts in all areas related to external controls as a result of his role at Office of Medical Policy. So, Dr. Concato, would you provide us with your perspectives about usage of external controls in terms of key considerations, appropriateness of using external controls given a disease condition, other appropriate considerations like cost implications, usage of real world data, and any and other considerations and comments that you want to talk about?
Oh, wow. That's a lot, Kanwaljit.
Yes.
I'll try
So
to
get started with the first
identify
one.
specific aspects of the topic you mentioned. And I'll also say thank you for the flattering comments, but I don't know that any one person is an expert in all things externally controlled trials. At FDA, we're pleased and privileged to have a very deep bench, I might say, to use a sports metaphor, in terms of having the requisite clinical, epidemiological, statistical, and other aspects to have subject matter experts to bring to bear, which we do with each and every application we see. But let me back up a bit and start with some very general comments, if that helps. First, I would say that FDA doesn't endorse any particular type of data source, say for an externally controlled trial, but even more upstream, quote unquote, from that, I want to be realistic and mention to the audience that the best chance of success for a new drug application would be to conduct a randomized controlled trial, not because that's the default and has been for some decades, but because of the strengths of such studies. To put it in informal terms, many more things can go wrong with an externally controlled trial. So in most situations, it's more efficient to get to drug approval with a randomized controlled trial when equipoise exists, et cetera. In certain circumstances, however, we have found that externally controlled trials do meet our evidentiary bar, which is one and the same for all types of studies. We do not have a lower bar for quote unquote real world data, real world evidence. So just as a bit of background, what we mean by an externally controlled trial, presumably many, if not most, are aware. But in a general overview of study designs, a traditional randomized trial and a randomized trial, even if there are variations on the theme, have a treatment arm and a comparator arm of some type. An externally controlled trial is essentially a single arm trial with a comparator arm that comes from an external source. So very generally, the two categories are either another, say, previously completed trial, which we don't see that often, or more commonly, what we now call a quote unquote real world data source. And in that latter category, examples include data from electronic health records, data from registries, possibly administrative claims, but as we go through this list, there are various strengths and increasing limitations. So in general, we have seen and approved drugs, if I say on occasion, I think that's fair. In fact, if we go back not that far, our regulations from decades ago talk about historically controlled trials, which could be viewed as a subset of external controlled trials. So I hope that in part addresses your question, Kanwaljit, and I'll allow my other panelists to, if I said anything wrong, please correct me, but hopefully that sort of sets the table for what we will be discussing. The only other point I'll make before others chime in is that for all stakeholders, especially sponsors, but all stakeholders, we did publish a guidance earlier this year on externally controlled trials, and it provides sponsors and other stakeholders with our current thinking regarding what I said, what circumstances to maybe consider an externally controlled trial, but also I'll be a little bit technical here and say that there's no magic number, but when we lumped and split, we came up with 10 different domains of comparability where the external control arm has to be comparable, i.e. similar to the treatment arm for us to even consider the possibility that the evidence meets our substantial evidence of effectiveness threshold. So I don't want to be pessimistic, but I want to be realistic. This is not a magic bullet. It's not going to work everywhere. And I think that RCTs in general are a better idea, but the issues are complex with regard to externally controlled trials generating valid evidence. And it's fair to say that we take it on a case-by-case basis, and we wouldn't be able to go through all of the considerations in this particular session. Let me stop there, Kanwaljit, and ask you first if that was helpful to provide some context.
Hi, Dr. Concato. This is Joe Hedrick here. Yes, that was very helpful, and I'm going to continue with
sure. Sorry, Joe.
No, that's okay. I'm going to continue with your baseball metaphor since we're in World Series season here. The bullpen with Dr. John Sharretts and see, John, if there's anything you'd like to add to this question.
No, I would say that I generally agree with everything that John Concato said. I think there are a lot of challenges in working in historical control. I do have some experience in the area, but not in the area of diabetes. And how we were able to operate or use an external control in that setting was, one, is that it was an exceedingly rare disease where there were literally like maybe a few dozen people with the diagnosis in the world. So a randomized controlled trial wasn't feasible. Number two was that we were able to build the applicant was able to build a very strong historical control, both by an unrelated population as historical control, but also by collecting historical control data from the patients enrolled in the trial. But even in those circumstances, like what made it feasible was that the natural history of the disease was, over the period of time of the study, was essentially no change or slight worsening, whereas the impact of the therapeutic was a large treatment effect. And I think that absent those things, it would have been very hard to interpret the data. In fact, with the same product, a little while later, we worked with a larger population. And again, it was a rare disease, but it was something where maybe there were 1,200 people in the United States who had the disease. And we required the sponsor to conduct a period of randomized control, which they followed up with an open label. And the open label data was very difficult to interpret. And they tried to do the similar kind of comparison to historical control, but we didn't have all those same features. I think the other challenge that with both of these situations was evaluation of safety. In the first group, where I think historical control was useful for evaluating efficacy, it was very challenging understanding some of the safety data. And we had the benefit of randomized control data from another population where we were able to understand some of the findings we had, because the background rates of certain safety issues may not be immediately discernible from a rare population. So I think the challenge with how does this apply to type 1 diabetes? Well, I mean, type 1 diabetes, I think it's feasible to conduct randomized control trials because it's not a rare disease. And I believe there are 64,000 people diagnosed in the US annually. In the world, I believe 500,000 people diagnosed annually with type 1. And depending on the therapeutic, like if there were a space where you're talking about a large treatment effect, then it would potentially be feasible, but we'd still have the issue of trying to understand safety data, trying to understand the magnitude of the treatment effect, and those are much more effectively evaluated in a randomized control trial.
Yeah, thanks. I think those are excellent points, and particularly on the safety side of things where that's a special challenge. I'll go just a little bit deeper into the bullpen to Dr. Pinsenstotler, see if there's anything that you'd like to add that we haven't covered so far.
Sure, and I'll be brief here. The other Johns did a great job. I'll frame what Dr. Sharretts said a little differently. I think there's a paradox with historical controls. The development programs that need historical controls, right, also carry the most regulatory risk. in other words, what the example John described to you is where you needed a handful of historical controls. You have a good idea about the disease, natural history. You know it stays flat. And this was a slam dunk product. The effect size was large. Okay. So oftentimes some of the programs, the folks that have, or the development programs or drugs or studies and so on, that would like to leverage historical controls, would like to leverage those controls because they have a smaller effect size, and that carries its own regulatory risk when we start borrowing people from other areas. Even during this this consortium, I met with a colleague who we were talking about how even matched patients could look very similar based on what we'd looked at, but except for those who were from a different country, and those folks, for some unexplained reason, progress faster in their natural history of disease. So I want to echo what John Concato said: is RCTs simply don't. Less things can go wrong with RCTs, but we do have to acknowledge that there's feasibility issues in some areas. I think just one more thing, Joe. I'll be quick. I don't think we can talk external controls, and I'm going to touch on my modeling background. I don't think we can talk external controls without talking MIDD. I think a lot of the things that can go wrong, John Concato touched on, like the 10 things we're looking for. You have to be able to look at those thoroughly. You need to model it and look at it all at once together. Okay, I think I think MIDD is really a pivotal technology for historical controls, and I at least the agency and through CPATH, we've been seeing a lot of that come in the diabetes space. And one last thing is I think this starts with a lot of these external controls start with very grassroots efforts to characterize the natural history. You'll see that we hear terms like clinical trial simulation, clinical trial simulator, and so on. And those are very low risk for regulators because, you're still going to conduct an RCT and we're still going to get the data in-house. And so that grassroots, these grassroots efforts by CPATH and colleagues and others, they start small, and I think with proper vetting and pressure testing, as they start to build up their use cases and context of use, they can eventually come to us as external controls. But that's a long, arduous process, of course. But I'll stop there. Thank you.
Thank you, Justin. I think this was extremely helpful to understand your modeling perspective and understanding the perspective from type 1 diabetes side of the things. I also wanted to pose this question to Dr. Massaro from Office of Pediatric Therapeutics so that we can get at least a little bit perspective about same issue from pediatric side of the things, specifically even neonatal side of the things also.
Is it on? Okay. Thank you. Thank you for that. And thank you for including me in this panel. I agree. I think a lot of the themes that I've heard over the last few days are going to be kind of echoed in comments here. I think we've heard that, there's not an all or one approach. And I think we've hopefully heard that we shouldn't be thinking of this as an either or paradigm, and we have to think about what the role may be here, and not that we're going to replace our RCTs with externally controlled trials. I think we heard good examples from Perdita yesterday about how some of these real world evidence approaches and things can be used to supplement and create a totality of evidence to support, product approvals. And I think that's the way we should be thinking about this. In neonatology in particular, our biggest challenge is, the neonatologists in the room know, is, we're the Wild West, as we've been termed many times. The variability in practice and the variability in care across centers, across providers, make it really difficult to think about, an externally controlled trial, an open label treatment, and being able to have interpretable data, as my colleagues from DDLO were just talking about as well. So I think as we think about these tools that are being developed and how they can be incorporated, I really encourage us to think about using everything at our disposal to put together a complete picture and and try to get as close to the truth as we can, and not say that this is the one magic bullet, as everybody's been calling out, because there probably isn't one magic bullet, but we need to figure out how these different pieces of information may work together.
Thank you, Anne. Yes, I think you go ahead. I think doctors. Yeah.
I just wanted to have one clarification though on what you said though is that we still have to remember what the regulatory standard is for new approvals, because you used the word totality of evidence, and I think that just rang a bell in my head, because that isn't one of the standards that we have. I mean, the standard for approval of a new drug in the U.S. is substantial evidence of effectiveness consisting of adequate and well-controlled trials. Now, the concept of adequate and well-controlled trials is traditionally was considered to be 2 or more trials, but there is language in the statute and guidances that talk about other pathways where one adequate and well-controlled plus additional evidence may suffice, and that's framed as confirmatory evidence. But there isn't really a standard of totality of evidence. You still have to have the one adequate and well-controlled trial in any scenario for approval.
Understood. Thank you. And I think Dr. Concato had raised his hand. Did you want to comment, Dr. Concato?
Yeah, I apologize if I'm interjecting or interrupting, I should say, but I am interjecting to just 2 points come to mind. I trust my FDA colleagues agree, and I think hopefully the audience appreciates the very high concordance of what you've heard from each of us. But and I'm deferring to the Office of New Drugs mainly, but please come to FDA early. Nothing worse than doing this, than going on this path and then sort of surprising the division with plans that are well along the way. And also, I want to emphasize in all of our real-world data, real-world evidence guidances, we really harp on the idea that the data have to be fit for use, fit for purpose, almost synonymous terms. The idea is that they have to be reliable, and that includes data quality, but they also have to be relevant. I think sometimes in my experience, which is not, drug or disease or division specific, the idea is that a database will be collected and there's an assumption that number one, it's going to work anywhere, or that number two, if it works in one context, it's certified, quote unquote, for another. Just like we don't use totality of evidence other than an informal way. It's, as John said, it's not it's not substantial evidence of effectiveness equivalent. The same thing here. We don't we take this on a case-by-case basis, and what might work for one research question, one drug outcome association, might not work for the next. So there's a lot to worry about. And the 10 items, they're not really items, they're 10 domains, and it's certainly not a checklist, but it's a good starting point for the conceptual thinking of FDA at this point in time. Thank you.
Thank you, Dr. Concato. I think at least this point is clear. It's no magic bullet. There are no, I guess, clear guidelines in terms of when is it appropriate or not. It all depends on a case-by-case basis. It depends on, I guess, as people were saying, what specific evidence exists. I think Klaus is on the mic. Did you have any comments? No.
It's working. Okay. So just a quick augmentation of what John, you mentioned, and what John Concato just augmented. Even though the term totality of evidence is not like a defined term with like domains and a full checklist and all that, it is not an uninformative construct to keep in mind when you're thinking about the everything that goes into the decision-making process for the approval, the review, and the potential approval of a product. I want to highlight the recent example in Friedreich's ataxia, where they still with a trial, so no question there. The trial that had to be done, but how a potential unanswered question about the actual difference between the treatment group and the quote unquote untreated population was augmented. And so in some forums that it has been called the augmentation of the totality of evidence by looking at data that was in the RDCA platform. And I wish Colin was still here to comment on that. And still not to replace the trial, but how that helped get a better sense for the separation of the effects between the treatment arm and the control arm, and how that helped inform a pivotal decision that came in February of this year. So that's an important aspect about the considerations about the evidence that needs to be shown in a very particular context, very well explained and put to use for informing decision making.
But just to make sure I'm not misunderstood, I was not endorsing totality of evidence. I think John and others would agree. At the end of the day, there has to be at least one adequate and well-controlled study and confirmatory evidence. If it's characterized in another way, informally, externally, that's fine. But am I correct, colleagues? That's our. In fact, we just released the guidance as CEDAR released the guidance on confirmatory evidence.
Yes. Yes. Yeah. Agreed, John.
Agreed. Thank you. And now we are actually really lucky to have EMA representation on this session today. So I'm just requesting if Dr. Bax, if you have any comments about all of what we've already discussed, and see if you are able to provide EMA perspective.
I don't want to disappoint you, but EMA obviously doesn't see it so much differently. On the other hand, so EMA has not published guidance. However, we have recently published a reflection paper on single-arm trials, highlighting also points we have just discussed. However, in pediatrics, we need to, there was an impetus from the pediatric space, at least in Europe, where there was a push for example, for extrapolation strategies. So it really depends, as you also mentioned, on the condition, on how many patients are available. I just looked at the recent PIP applications for type 1 diabetes, and all the proposals actually. I think there was one proposal looking for external control, but here also there will be a push for active control. And then we had the other proposals recently were active control and placebo control trials. So in neonatology, as you have said, it will be hugely difficult. And therefore, we are looking forward to also all the data you are collecting. From the pediatric oncology space, we have had recently more proposals for also external controls. And here I just wanted to say that when looking, and they even came from specific registries, but however, when we then looked at the quality of the data and the granularity of the data, the conclusion was actually in most recent cases that these data were not sufficient as a basis for external control. So this community is now looking prospectively in also enhancing what registries are collecting, because registry is not doesn't equal registry. So also in Europe, we are now looking also at a framework for quality of data submitted, and that is also the crux in the end, in the practical approach. Thank you.
Ralph, if I know the drug you're talking about, FDA reached a similar decision with regard to an externally controlled trial in the oncology space. We need not discuss it, but yet for the same reasons. So I think, as you said, there's high concordance between the two agencies.
Yeah, I'm glad to see that there is definitely a concordance in terms of what the thought processes are. And after having, I guess, having heard from different divisions of FDA, EMA, I also wanted to refer this question to our industry colleagues who are on the call today to understand what do they think. I know, Victoria, you have been very active in doing clinical trials in neonatology. So I'm really very curious to hear from you. What are your perspectives about appropriateness, when it is not, pros, cons, and whatever your thoughts are?
Absolutely. Thank you for that. So I think from the perspective of a neonatologist, historical controls is almost ancient, because I think what's critical, it tags off of what Anne said, is the practices have changed so dramatically over the last 30 years. If I were to consider historical controls from the time I was an intern, I'd be perhaps exacerbating or achieving an abnormal treatment effect, unrealistic treatment effect. So I think assignment of those controls has to nearly be temporally next to or contemporary, which means we need the ability to be able to access databases almost in real time so that those subjects are truly matched. So that's that is one point. The second would be that if, a magic wand might be that using a previously done clinical trial close, in close proximity to, so for example, a phase 3 trial on the tail of a phase 2B, using those subjects as the control might be one approach. But clearly, I've certainly reached out to Susie McCune in the audience about, Susie, how can we execute this trial more quickly using a contemporary external control arm? And she's like, not yet. And the downside is we don't have an approval. So we don't have a previously approved drug in the space. So there's so many things that are stacking that make it a challenge for us. That doesn't mean I don't have a magic wand in terms of what I would like. And if I were to have a magic wand, I would choose the ability to enroll subjects from an existing contemporary, temporally associated population, real time. I think that's where I see a real benefit, and where that would be something we could do.
Good to know. Thanks, Victoria. I think the point is populations need to be well matched, especially if the standards of care are changing rapidly. It's extremely important to be staying current to, where the population lies. Tom, I really wanted to hear from you also. I know you've had some experience, even though I would say some is kind of within parentheses. I know that pretty successful experience in using external controls in some of the work that you've done. Wonder if you want to comment a little bit about it.
Maybe just a couple of gems to plant with from my industry colleagues, and then I could jump into the specific example. So number one, I think I share the health authority colleagues' opinion that if patients are plentiful, this is not a relevant strategy. So just be mindful of that as you think through your respective programs. Secondly, I've found the most success in having a productive conversation with health authorities when we had a very thoughtful approach towards the proposal, not just going in and simply asking, can we use an external control arm, but rather really trying to defend your rationale for how relatively fit for purpose the data set is, how relatively contemporary it is, and how well does it match current standard of care. Where you plan on conducting your trial? Do you have overlap with where that data was sampled from? So on and so on. So and also a thoughtful assessment on how you would approach your statistical analysis plan if you were to propose such an approach. So my advice would be to be thoughtful. And thirdly, it seems our discussion thus far has been almost like a binary conversation. Should we or shouldn't we? But there's potentially variations on the theme as well, where one might consider partially synthetically controlling a study. And that's the area where we found some recent successes. This was a study in a population of infants suffering from a rare complication of prematurity. And sometimes in this business, I'll take all the luck that I can get. And as it turns out, through pure serendipity, a sponsor company that I think very highly of had conducted essentially an identical study with almost identical inclusion-exclusion criteria with a very similar molecule in the same patient population about a year before we planned to, and published the data in a high-impact journal as well. And I anticipated that this company had a commitment to data quality that was, I suspect, no different than our commitment to data quality. So we proposed an approach where we would use this as a partial synthetic control. We did prospectively enroll a handful of patients in this patient population. So we had a way to sanity check the adequacy and comparability of the data, but we nonetheless primarily the primary patient population for the control population was from this alternative trial. While I was pretty pleased with the feedback that we had from a couple of key global health authorities, what I also learned through this exercise, bless you, was that this can translate to an operational success within a rare patient population as well. In my experience in rare pediatric populations, consent rates, attempted consent to actual consent is not very good. And I know this is true as I've spoken to countless colleagues in academic medicine in their own clinical trials as well. Sometimes it's hard for caretakers and mom and dad to be magnanimous when there's a chance that their child could be randomized to standard of care. In this particular case, we had an extraordinary rate of success in a first-time attempted consent to actual secured consent. And in this particular population, which is a rare complication of prematurity, as I indicated, we enrolled 120 patients prospectively in 9 months during a pandemic, which was an extraordinary feat from my experience. The team did a fantastic job with the trial, and we're now registered globally in 29 countries, and I have one more to go. So hopefully an opportunity to close out this chapter and hopefully a good solution for preemies suffering from this particular condition. But I would say this is a very, you heard this from many health authority executives. There were a number of things that had to come together for this to even be possible. So this, I don't think, is representative of a default position, but in this particular circumstance, it worked out.
A follow-up comment, Tom. You said that there was between attempted consent and actual consent, there was a very short time interval, and you were able to enroll large number of patients in a short period of time. Was that because the patients knew that this is effectively kind of a single arm trial and they're all going to get the...
Yeah. So just to clarify a little bit, it wasn't about the time interval between, it was the success rate, meaning that I think it was almost 95% of patient caretakers that we attempted to consent actually consented, which is not something that I experience very often. So it's difficult for me to say this was directly attributable to the design of the study, but the design of the study and the relative probability of receiving active was made very clear to the caretakers in the consent process.
Great, thanks. Dr. Allen, I'd like to go to you here. I think having heard a number of the challenges outlined by industry and the considerations from both the EMA and FDA, what do you see? How can IACT or how can these various communities maybe come together to overcome some of these challenges?
Yeah. So, one of the things, and I'll use a couple of examples here. I think there's often a lot of wishful thinking or imperfect understanding between the different communities. I don't know how many times I've heard people from FDA talk about things in just like everyone's been talking here, and I typically agree with them. And then I talk to colleagues in industry who may or may not have a similar sort of perspective, which is, it's not one size fits all. This is something you have to have some very careful thinking about. You can't start thinking about real world data as being a source of doing a trial
because we did the feasibility analysis and we saw we didn't have enough patients to do a double-blind study, so we got to do something. And what about doing real-world data? And here's a data source we could use, and we'll convince ourselves mentally that it happens to work. That's not a good approach. I think the best approach is going to be to have conversations early, to look at the different possibilities. We've had other conversations about extrapolation. Think about in the pediatric space. Okay, is this something where extrapolation could apply or not? If there's gaps in the data, is it possible to collect data on those things that would make it reasonable to use extrapolation? If there's too many questions around that, then are we going to need to do a separate trial? Can we potentially, what are the potential sources of control that are out there? Can it be a double-blind placebo-controlled trial? If we can't necessarily do a double-blind placebo-controlled trial because of ethical reasons or other things, what are other approaches that could be used? Could we actually generate our own real-world data leading up to this by starting a registry that's high quality to the standards that are required by the FDA guidances in terms of those meeting those 10 criteria. And then when we're ready, we actually can proceed with the study. But all of this takes communication and all of it takes time. So, one of the things that for me has always been important, we don't ever get time back, but as you proceed with time, the number of opportunities, the number of possibilities that you have generally tends to narrow down. So you can box yourself into a corner by delaying and delaying and delaying, and finally you're left with, okay, our only option is to do this, and it's not a great option. And the agencies look at it and they say, "Are you nuts?" Type of thing. And I think we have to acknowledge that happens, but we can prevent that. We can start the conversations early. We can have a real meaningful discussion about what's the science, what is out there. There are going to be times that people are going to disagree. I mean, the FDA and EMA agree most of the time, but not 100% of the time. And I think the same is true if you get any two scientists together. They'll agree much of the time on some of these things, but there's whole careers made of battles over particular topics. So don't be unrealistic. We're people, okay? And I think having those communication, having those contacts, exchanging ideas, there's a lot of. I used to spend a lot of time when I was at Lilly. I'd say to our regulatory scientists and the teams, I said, well, why don't you go ask the FDA this or ask the EMA this or something? And they, one reason or another, they didn't want to do it. And I said, I'm not a mind reader. I can't tell you what Lin Yao thinks. I can't tell you what EMA thinks on these things. There's a reason to have those conversations, and it helps to advance the process. And I think all of that, in my mind, we need to keep those basic things. It's not rocket science. It's basic human dynamics, human interactions, and cooperation, collaboration. And remember, the end goal here is to try and benefit kids. So,
can I follow up on something that AJ said? So you gave a very eloquent introduction in your comments about what not to do and what you wouldn't advise folks to do. But this is really directed to the FDA and EMA colleagues on the panel. I suspect you've all heard more than once from a sponsor that the traditional approach is not feasible. So could you give us maybe some advice, not necessarily guidance, but is there a standard by which feasibility or lack thereof is gauged? Is there a patient epidemiologic threshold, or is there something else, or is it just another case by case thing?
We'll go to Dr. Sharretts first, and then Dr. Bax, if you care to add.
Yeah, I mean, I think I can address this. I mean, I think the thing when you're approaching the trial is I heard some of the challenges that you talked about with recruitment, and but it's really important that that you communicate to potential participants what's actually going on. If a a therapeutic is being evaluated in a trial, it's because we don't know whether it works or not. So if you tell me that participants are more likely to be enrolled because they think it's a 4-to-1 randomization, they have an 80% chance of getting the therapeutic, what I'm hearing is that somewhere along the lines, people have gotten the impression that this therapeutic is effective, and you don't know that. And I would say that you have to be able to explain to people who are considering enrolling in a trial that if they don't enroll in the trial, they are getting standard of care, and there's a 0% chance that they're getting the therapeutic. And if they get the therapeutic, there's a real possibility that what they're getting is not an effective product. So even if you have an 80% chance of getting it, it may not be beneficial. And if those things aren't spelled out in the informed consent and explained to the patients, well, yeah, there are going to be challenges with enrollment. But more importantly is if we don't conduct the trials, if you don't conduct a trial that demonstrates the effectiveness of the product, then you potentially are putting on the market a product where we don't know whether it works or not. And I mean, for me, I would hate to say that I'm taking a medication for a disease and there's a 50-50 chance that it works. I mean, that's actually worse. And that's why we conduct trials. Yeah, enrollment is the element of randomization doesn't mean that some people get therapy or no therapy or placebo, but it's important. If there's available therapy, then maybe that needs to be a background therapy that's offered to everybody. If there are no available therapies, it needs to be really clear that the therapy being evaluated is experimental, and we don't know whether it works or not, and that's why the trial's being conducted. So, I mean, as far as feasibility, I mean, I think the things that make something infeasible is, like, there literally aren't enough patients to do a randomized controlled trial. So the trial could only enroll 10 subjects. If you have enough subjects that you can do randomization, it's really the best way to find out whether or not the therapeutic works.
Dr. Bax, I get the.
Yeah, I just wanted to comment on part of that, because it's important to remember we're talking about pediatrics now, and I'm going to take my clinical trialist hat off somewhat and put on my bioethicist hat, because according to the Subpart D regulations, if you don't have any idea about whether or not there's a prospect of direct benefit, you really can't do the trial in pediatrics. So we have to have some prospect of direct benefit. Now, I'll grant you, it may be very small, okay? And in rare diseases that are where you have a serious ill child, the 50-51 category has a sliding scale in it. So that very small prospect, given the risk that you're associated with, makes that very small prospect okay. But there has to be some reason to believe, to do the trial, that it might benefit these patients. It may be very small. So I agree with you generally on what you're saying, but we have to keep that in mind as well.
Great, Dr.
Sorry. Yeah, sorry. I just wanted to add on to that. I appreciate you bringing up the pediatric regulations here. And for sure, there has to be a prospect of direct benefit. But I also want to emphasize the importance of the phase 3 trial. In neonatology, especially, we live in a space where a lot of what we do is off-label because we just believe that it may work with very little evidence. We've had many examples, and even very recent examples, where very promising interventions and drugs, looked very promising in phase 1 and phase 2, and just erythropoietin was one of our biggest examples, where there was so much enthusiasm and a lot of use clinically, and one of the best conducted trials that I've seen and had the pleasure to be involved with, I think really answered that question for us. So I think that all of the points that are being made are true. And I would argue in neonatology, these trials are incredibly challenging. But I have a hard time saying they're completely infeasible, because we have had trials successfully conducted. And I think all of these things that we're talking about are ways to optimize the way these trials can be done, the way they can be designed, the way they can be made more feasible. But I worry about going down the road to say that, we can't conduct trials in some of these spaces that will really get us the answers that we need.
Thanks. I want to hear from Dr. Bax, and then I think we'll go to Betsy on the line to hear her perspective from the patient side of things on some of these issues and other issues that she may wish to discuss. But Dr. Bax?
Yeah, Tom. Yeah, indeed, we often hear this argument, and then we just ask back, what do you understand by not being feasible? Is it, could be all sorts of reasons why it is not feasible for a certain applicant. In some cases, we might know actually from other programs that it might be feasible, or we need to talk to experts comprising patients, clinicians, and to discuss their view, because as a regulator, we need this input in order to push. And then I'm coming now also from a pediatric regulation point of view, because this situation within the pharma regulation, as you know, is a little bit more unique. So we do need to push. We do need to come back to the applicant, explain to us exactly where are your issues, and then to explore how to manage to overcome these issues. So not being feasible would not be an argument for us.
Thank you. So Betsy, I think you've heard quite a bit of the discussion here. I'd invite you to offer your perspective from a patient side of things, but also would like to hear from you. What are some of the things that people could do when they're thinking about trial design to make them more attractive and to maybe increase the feasibility, as it were?
Yes, thank you. I have a couple of thoughts that I wanted to share. Just overall looking at, and of course, I am extremely biased as I come from the patient advocacy side of things, but engaging patient advocacy early and often in any of this work is so critically important to find out truly what matters most to the population you are studying, and really look deeply into their experiences. And looking at, when you talk about enrollment, and especially, I don't have as much experience, although we're working through that. A lot of our families do engage in pediatric trials later on for a lot of the subsequent diagnoses that come from HIE. But certainly in the acute neonatal period, there's a lot of complexity. Especially with our population. We have families who have participated historically in nearly every single HIE trial, and have a lot of expertise that they have shared and feedback along the way. And in particular, Anne mentioned the HEAL study, and we had almost half of the cohort just naturally find us. So I think a lot of the learning that we have gathered over the years from this population really talks about the complexities and understanding how you can back up and start early with trying to look at what barriers exist and how to decrease them. And so, this isn't going to be necessarily, although I'm sure there is some science behind this, and if there isn't, there needs to be more about, how families in the midst of an acute setting, which is inherently traumatic, in particular, if you're looking to enroll quickly, like we do in HIE, within, 6 to 12 hours of birth, where, maternal, health can be impacted, where there's variance in family dynamics. It seems like there's just a lot of complexity, and how we can help families make informed decisions about these trials, about, clinical trial participation in general, about what could be gained from these for their child, and doing so in a way that maximizes that state of cognitive processing for these families, and taking it really from an approach of like a health literacy standpoint, multisensory, having things that are in first language of the family that you're working with, and then ultimately connecting to longitudinal support for these trials. And that's where I think I will evangelize continuously, partnering with patient advocacy. So, like I said, our community in particular, we have over 9,000 families today connected worldwide, across many different ages and outcomes and locations. And so we have built in support as we've grown, with a social worker, child life, lots of the things that families will face with medical complexity along the way, and seeing some of the barriers of participating longitudinally. I've seen, and I know you all know the statistics of attrition and things like that in the neonatal space, and how do we work together to prevent that and take really a holistic patient engagement and education strategy behind that. So those are just some of my initial thoughts that I wanted to share, and would love to hear any further discussion on those as well.
Thank you, Betsy. Just wondering, I think AJ, did you have any comments?
Yeah, I just wanted to comment on something she was saying and put it in perspective, because one of my interests is the bioethics of trials. And when you look at Subpart D and you look at the history of it, what they tried to recreate in that, this is the regulation for around informed consent, when we can do trials and what the conditions are in the pediatric space. Subpart D is constructed because in a way that's trying to help mimic what the concerns of families are. When you have somebody that wants to do a clinical pharmacokinetic study or something, and they say, we're just going to do a single dose and then we'll look at safety and things around that. And there's no prospect of benefit for that individual patient that has a chronic disease. Why in God's name would any parent put their kid in a trial to go through that? Okay. The regulations actually say you shouldn't do those studies. In most cases, unless there's some sort of benefit to that single dose. And it's important to recognize that. And really ultimately the question in many cases in terms of recruitment into pediatric studies and involvement comes down to how well do we do informed consent. I just want to note that oftentimes we have multiple pages of informed consent. There is, under the new informed consent guidelines, one of the things they talk about is key information. Unfortunately, the lawyers gotten a hold of that, and it's often much longer than it needs to be. But you want to know what key information is? It's pretty much the same thing. And I say this as somebody that was involved in providing input on the regulations. It's basically the same thing as a clinician would do with a patient bedside. These are the potential benefits you might see. These are the potential risks. These are concerns you need to think about long term. Maybe this is a gene therapy study and you can't have any of this sort of therapy in the future, that type of thing. It's not meant to be pages and pages. It's meant to, maybe be a paragraph or two that lays out these things in the way that a clinician typically would. I say that because the people that understood it best were the folks that were clinicians that were talking about it. Whether you have other stuff in the informed consent pages, that key information piece needs to be like that. And it's the reason it needs to be like that is that's the way that people normally can get information and understand it and be able to make a decision. But think about the patients, think about stuff from their perspective, what they need. Much of the reason people don't recruit into trials is because we haven't done that. Think about if you were the parent of the kid that's potentially going into this trial. So thank you.
Thank you, AJ. I do see that we have at least a couple of comments from the audience. Do you want to go first?
Yeah, Matthew Ellenwood, National MPS Society. I'm sorry to hijack this session. I should have spoken at the last one.
No worries.
The idea that we need control groups, and this is continually expressed to us by CEDAR when they come to meet with the MPS Society, that we need placebo control arms or rescue arms for these rare neuropathic diseases is an obscenity. It might as well be putting a kid on a marble slab and opening their throat. You are consigning them to a lifetime of
either no response or extremely advanced disease. We know what causes these diseases in the case of soluble protein defects. We know how they respond biochemically. And guess what? We don't have gaps in the natural history. Neuropathic MPSs are uniformly fatal, approximately 15 years of life. With an incidence of maybe 20 births a year, requiring us to build new databases that will get you the data you want, that's unconscionable. The FDA needs to figure out a better system to get to advanced or accelerated approval on biomarkers. That is the only answer. Asking these communities that have been so traumatized to help foot the bill to get there, there needs to be a better way. And it needs to be done quickly. Hundreds of children are born every year, and they face years up to decades inchoate, recumbent, tube-fed with aspiration respiratory rescue. We need to figure out how to do it better. And it's not filling in the gaps. We know these are uniformly fatals, and the parents know the risk they face, and they're very willing to sign up. Thank you.
Thank you, man. I think, Judy, you had a couple of comments.
Yeah, I was going to return to some of the conversation that we just had about the challenges in recruitment to neonatal trials. I was going to make a comment about our informed consent. So thank you, AJ. At my institution, our typical consent form is between 26 and 34 pages long. The first 2 or 3 pages has what the family needs, and then they just drown in all the institutional language to indemnification and protecting the institution. And by the time they're done, they have forgotten why we've talked to them. And I really, really think that this is an area for some advocacy and for us to realign ourselves into something that families can actually understand what they're signing. I think we've defeated the whole purpose of informed consent because they can no longer be informed when we have a consent form that's long. The other thing I wanted to mention is there was a comment about longitudinal follow-up and loss to follow-up. That's on us. We do a terrible job of staying in touch and communicating with our families after they do agree to participate in our clinical trials. Where's our return of results? Where's our sharing of information in an easily digestible format? What we've learned, what we've found, why this is important to them, what other information has come out in the literature that was subsequent or parallel or unrelated to the trial they participated in. We as a community do a terrible job of keeping families engaged in the research that we are doing that their child, that we couldn't have done without their consent. And the last thing is that we really could do a much better job with education. We're in this room kind of speaking to the choir, right? We talk to one another. We're all on board. But when families, particularly NICU families, and I'm a neonatologist, and I had a NICU baby, and he fortunately got compassionate use of a drug that was still in trial development, probably the only reason why he survived. But, who do these families go to when they are under stress, which they all are when we approach them? They talk to the bedside nurses. INC has addressed this to some extent, and we need to do much more to educate the general neonatal nursing community about the importance of research. And we need to get information out to the primary care pediatricians. If they have an older sibling, they're going to call their pediatrician to say, I have a baby in the NICU, and what should I do? And I think a lot of our community doctors are not well informed about the importance of research. And then they go talk to grandma. So we need to have information for the general public. And I think these are all very important things that we could tackle together. And I don't think we do very well right now. And it's hindering our ability to recruit, enroll, and follow up these kids.
I agree, Judy. Thank you so much for your comments. I'm pretty sure there's a lot of work that we still have to do. And sure, we'll get there. I was informed that Betsy had a comment and she had raised her hand. Betsy, do you want to go?
Yeah, thank you. I wanted to follow up on a couple of points that have just been made because, when we talk about the informed consent obviously being onerous, to say the least, and the overview kind of distilling this ability to have a piece of literature. That is still what I am seeing in practice. I am going back with the sites that we're working with who send me what they have. These things are written so above and beyond what the average person is able to understand, even distilling these, very lengthy patient consent documents. And so again, I think that there just needs to be an emphasis on what that level of building, considering literacy in general, and then considering health literacy and backing up a few steps. Because I just continuously see that it's trying to distill these very, very complicated concepts, and it's still missing such a huge mark that I fear that we're just creating more issues than what could be really beneficial, again involving the patient community to drive that in conjunction with the study sponsors and the site.
Thank you so much, Betsy. I think we've gone way above the time that we had for this session, which again turned out to the last session of this meeting and the day. I really wanted to thank you all for your time and for being here. It's really good to see you all here, being here with us and listening into the sessions. I really hope they were informative. But I would also like to say thank you to all of our panelists for your helpful comments. Do greatly appreciate it. And most importantly, I wanted to express my sincere thanks to my CPATH colleagues, Joe, Klaus, Dan, Colin, Colleen, Christine, Olivia, Karen, Sarah, Krista, Brianna, Aaron. Apologies if I'm missing anybody here, for all of your tireless work in making this event possible. Since months and months, we have been working on arranging all of the logistics, agenda items, panel questions, scheduling panel planning calls, all of the logistics involved with getting the setup ready here. It was an unbelievable amount of work that you guys did, and you have my sincere thanks for getting it done. I would also like my colleague Klaus and Joe to offer any comments that you might have.
Joe, do you want to go first?
Yeah, I'll just add one comment, and then I'll have Klaus call it out. I think one of the things that I found really interesting here that we're going to need to think about is, and I think type 1 diabetes is an example of this, but not the only example, as we think about intervening earlier in disease before the onset of clinical symptoms. We've talked a lot about the screening of challenges. So even in disease, the challenges of screening rather, so even in diseases that aren't rare, when you look at how many people are identified by a screening paradigm and how many patients you actually have to approach, and they're not even patients at this point, healthy individuals that you're going to invite into a trial, you're going to be at very vanishingly small numbers approaching what you would see with many rare diseases. So I think as this paradigm changes of treating disease to intercepting disease, if I might, I think that's something that we'll need to think about in terms of some of the discussions that we've had all across this meeting. And with that, I'll turn it over to Klaus.
Right. So yeah. So thanks for being here. First of all, the one take-home message that I want everybody to walk out of this room with is the notion that together we're going to make the changes that need to be made, and we're going to advance the paradigms that need to be advanced, without reducing the scientific and regulatory rigor. And the FDA deserves a ton of credit for being so visionary in establishing these public-private partnerships to make sure that these conversations happen. I know that it's very easy to criticize governments, but that's not what we're here to do. And if anybody wants to keep criticizing government, there's many forums for that. This is not one of them. This is the forum to make sure that we work together to make sure that we do right by the people that are in need and deserve both efficacious, but importantly, safe medications. And so just want to make sure that everybody leaves the room with that message, and that together we're going to be very strong to make sure that all these populations are helped and get the medical products they are waiting for. So thank you very much and safe travels home.
Thank you. Thank you. I forgot to thank Cecile for her help too. Sorry. Thank you. I hope to see you all again next year in our follow-up meeting.
Yeah, exactly.